Oh, come on. Let's- Okay, can somebody confirm you can hear me?
Yeah, I can hear you.
Hear you. Thank you. My nice mouse isn't working well today. Let's see, what lesson are we on? Three, starting chapter 4. I think we did start chapter 4. Okay, before we begin, are there any questions?
No.
Thank you. All right, let me share my screen. Find button up here, and I'm not finding it anymore. There we go. So I have that we had talked last time about the endoflagella. And I remember talking about the endoflagella. Any questions about the endoflagella? Let me get this out of the way. It's a flagella found inside the cell wall, found only in spirochetes. All right, let's talk about fimbriae. Fimbriae are shorter, straighter, and thinner than flagella. Here's a cell with multiple yellow fimbriae. Fimbriae are used for attachment to something, like the cell, if it's on something, can attach to it with uh, these fimbriae attached to the surface. And they are important, like on human pathogens. The fimbriae allow the human pathogen to attach to human cells. Fimbriae are not used for swimming, so the cell cannot use it for locomotion. Any question about fimbriae? Nope, went the wrong way. Pili, also known as sex pili, are longer than fimbriae, and they are hollow, and they're used to transfer DNA from one cell, this cell here, into another cell, such as this cell here. Pili are only found on certain cells, which are usually called F plus cells. We'll talk about that later. Any question about sex pili? All right, let's talk about the prokaryotic cell wall. The prokaryotic cell wall is complex and it's a semi-rigid structure. It is responsible for giving the bacterium its cell shape. The cell wall does protect the cell and prevent osmotic lysis. If a bacteria cell were to lose its cell wall and it were to be in an environment where there's more water outside the cell than inside the cell, water would move into the cell and the cell membrane would expand. And the cell membrane would continue to expand until it eventually popped. And then that would kill the cell as the cytoplasm would leak out of the cell. The cell wall prevents osmotic lysis by allowing the cell membrane to expand up to the cell wall, trying to show that here, And then the cell membrane can't expand any further because the cell wall prevents it from expanding. And that's the main way that the cell wall protects the cell, keeping the cell membrane from expanding and then popping, bursting. It can, act like a wall and protect the cell from foreign, I guess, objects or entities. But you got to remember a bacteria cell is really small. And so the cell wall frequently doesn't act as a wall to protect the cell from, I don't know, like a branch falling on a cell and then puncturing the cell. Obviously, a branch would be much, much larger than a bacterium. So the bacterium would probably be fine. The cell wall is located outside the cell membrane. And even though it's located outside the cell membrane, we do consider the cell wall a part of the cell. The cell wall is the last part of the cell, meaning anything further out of the cell, like the capsule, is considered external to the cell. The cell wall can contribute to the pathogenicity in some species, like the Mycobacterium species. The cell wall definitely contributes to their pathogenicity. The cell wall also has receptors for attaching to their host cell. And we'll talk a little bit more about that later. And then the cell wall of bacteria is made up of peptidoglycan. This is not true of the cell wall of archaea or the cell wall of plants, protists, and fungi, meaning any eukaryote does not have peptidoglycan. Archaea also generally lacks peptidoglycan. Peptidoglycan is a molecule found only in bacteria in their cell wall. Peptidoglycan is a polymer of a repeating disaccharide, NAM and NAG. Let me blow this up. And what you have is the NAG and NAM are sugars, and they just repeat one after the other in a row of them covalently linked together. So here we have NAM. Come on, my mouse died. There it is. NAM, NAG, NAM, NAG, NAM, NAG, etc. Uh, NAG stands for N-acetylglucosamine, NAM for N-acetylmuramic acid, and those names are probably as familiar to you as NAG and NAM, so you can call them either way. The peptidoglycan forms a lattice surrounding the cell. Now, this row of peptidoglycan is hooked together by a covalent bond, hooking the NAM to the NAG and then the NAG to the NAM, etc. But the peptidoglycan row has a row on the side of it and another row on the side of it, and then a row below it. And actually, this is a, what do you call that, a plane? And there's another plane of NAG and NAM underneath it. And the planes are hooked together. The NAG and the NAM rows are hooked together by the short peptide polypeptides. What happens is this row of NAG and NAM is hooked to this row of NAG and NAM by this short peptide. And then this, come on, row, of nag and nam is connected to this row of nag and nam by this short polypeptide. And this peptide is linking this peptide to that peptide. So that they are linked, this one to that one, but through this peptide and then that peptide. And then this peptide links that row to that row and this row to that row. So the lattice of peptidoglycan forms the cell wall. And remember when I told you there was a layer of peptidoglycan? When I'm talking about one layer, that would be one plane of the NAG and NAM chains. So that would be this one, that one, that one. That would be one layer. And then the next layer would be this layer here, which is another plane of them. And then there's a third one. And if you remember, I did mention that peptidoglycan is normally, even in the gram-negative cells, 3 layers thick. And in the gram-negative cells, they're usually between 3 and 20 layers of peptidoglycan. In the gram-positive cells, they're more than 20 layers to more than 100 layers thick. Any question about peptidoglycan? Penicillin actually inhibits the formation of the cell wall by interfering with the linking of the peptides to each other, like this one here. Penicillin actually, when the cell wall is being formed, the NAG and NAM will be laid down, and then later the peptides are attached, and penicillin will get right here and then prevent this linkage. And then that'll create a hole in the cell wall right here. Come on, mouse. There we go. So there will be a hole in the cell wall here, and then the cell membrane can protrude out of this hole. And then water can move into the cell, and the cell membrane will just protrude further, sort of like blowing a balloon up in a box that has a hole in it. The balloon will protrude out of the hole. And you keep blowing the balloon, eventually the balloon will pop, not where the balloon is protected by the box, but where it's expanding out of the hole. And that's what will happen to a cell when penicillin uh, damages the cell wall. Any question about any of that? All right. So let's look a little bit more at the gram-positive bacteria cell wall. It has a very thick layer of peptidoglycan. And it's more than 3 layers. It's only shown 3 layers here. 1, 2, 3. This would be the minimum size, actually, of a gram-negative cell. But the main molecule in the bacterial gram-positive cell wall is peptidoglycan. There are some other molecules in the gram-positive cell, such as teichoic acid, which runs down the peptidoglycan. Gunk, gunk. And it actually helps link the peptidoglycan together. So it gives support to the cell wall. We also have a lipoteichoic acid, and it has teichoic acid in this part of the cell wall. And then in this part of the cell wall, it has a lipid attached to it. And the lipid will go into the cell membrane. Any question about any of that? So teichoic acid is definitely the main other molecule besides peptidoglycan in the cell wall. Teichoic acid is thought to help regulate the movement of cations, meaning a positive ion can, from this side of the cell, come into the cell wall and then move down the teichoic acid and then eventually move into the cell. So the teichoic acid helps regulate the movement of cations. However, cations are not needed, do not need the teichoic acid to move into the cell. A cation can move right through the cell wall. And that's actually true of any molecule that is, what's the word I'm trying to say? Dissolvable in water. Water can move right through the cell wall of a gram-positive cell. And that's because the peptidoglycan is, what's the word? A polar molecule. And water's a polar molecule. And then a cation is actually an ionic molecule, and it can dissolve in water. The gram-positive cell wall also has polysaccharides on it that provide androgenic variation. And so we can classify different species by their androgenic variation. And you may have heard of something like E. coli 0157H11. Those 0157 and H11 are different antigens on E. coli. And they're from polysaccharides in the cell wall. Any question about any of that? All right, let's take a little greater look at the gram-negative cell wall. It does have a thin peptidoglycan layer, and outside of that peptidoglycan layer, there is a lipid bilayer that is part of the cell wall, and we call it the outer membrane of the cell wall. This layer of the outer membrane is primarily made up of phospholipids, the same molecule that we find in the cell membrane right here. The outer bilayer of the outer membrane of the cell wall does have phospholipid in it, and that's these, well, that's a phospholipid icon with the yellow head and then the blue tails. And that's one of the main molecules, if not the main molecule, in this outer layer of the bilayer of the uh, outer membrane of the cell wall. But there are other lipids in it, and that's that big globular blue molecule. That's lipid A. Attached to lipid A is an O polysaccharide, and that sort of looks like a hair coming out of that blue blob of lipid A. And together, lipid A and the O polysaccharide, we call that a lipopolysaccharide. That is a molecule inside the outer membrane of the cell wall. You can also find porin proteins in the outer membrane of the cell wall. A porin protein is important because they make a channel that allow any molecule that should come into the cell to traverse the lipid bilayer of the outer membrane. And the porin protein allows many different molecules to come through it. That differs from the cell membrane. If we have a molecule that cannot move across the lipid bilayer of the cell membrane, it is moved across the lipid bilayer of the cell membrane by a transport protein. But there's one transport protein for each molecule that the transport molecule moves, meaning there's a separate transport protein for fructose and a separate transport protein for glucose and a separate transport protein for 20 different amino acids. So there's 20 different transport proteins just to move the amino acids across the cell membrane. And all of these molecules can move through the porin protein because it's just a channel protein, allowing certain molecules to move through the outer membrane. And something that should move into the cell will move into the cell wall and then through a transport protein into the cell. But if it's going to leave the cell, it needs to leave the cell through a transport protein through the cell membrane and then leave through the porin protein in the outer membrane of the cell wall. Any question about any of that? Also in the cell wall, we can find a lipoprotein as well as proteins. The lipoprotein, the lipid portion of the protein, will be in the outer membrane of the cell wall and in the cell membrane. And then the protein portion will be in the peptidoglycan layer. Any question about any of that? Let's see, have I mentioned all the molecules? The periplasm is the region of the gram-negative cell wall that is between the cell membrane and the outer membrane of the cell wall. So the periplasm is the region where the peptidoglycan is found. And remember, in a gram-negative cell, we have at least 3 layers of peptidoglycan, but it's thin. There's not more than 20 layers of peptidoglycan. Any question about any of that? All right, so this is showing you the gram-negative membrane in a little more detail. So there's the peptidoglycan layer. There can be proteins running throughout the cell wall, such as this lipoprotein. And there's a lipid portion, which I'm not sure where the lipid portion ends, but it's probably going to end in the cell wall because I'm not seeing it here. That's a porin protein. That's all protein. And then this lipid portion, lipoprotein, can also run into the cell membrane. Here it looks like a protein running through the cell wall. Lipid A, that's the important molecule in the cell wall. Actually, it's in the outer membrane of the cell wall because lipid A is an endotoxin. An endotoxin is a toxin, and toxins are substances that are toxic to us. So all gram-negative cells that have a cell wall contain lipid A, and that means anyone who has a gram-negative infection that has a bacterium that has a cell wall, they will be exposed to a toxin, lipid A. Any question about any of that? And I see an arrow saying that there's a peptide, but I'm not seeing the peptide there. That would be like a protein. Obviously, there's a protein linking peptide linking this nag and nam chain to that nag and nam chain. But I don't see one there. So I'm not sure what they're pointing out there. All right, so on the gram-negative cell wall, we have lipopolysaccharide. This includes lipid A, lipoproteins, phospholipids. The gram-negative outer membrane of the cell wall can help protect the cell. It can protect the gram-negative cell from complement proteins. These are proteins that an animal makes to help defend themselves against a foreign microbe. we'll talk briefly about these later in the term. And then antibodies I've already talked about. Um, the antibodies will have a harder time attaching to the gram-negative cell because of the outer membrane. And then there's certain antibiotics that will not get through the outer membrane of the cell wall. The main one, and the only one I'm going to talk about, is penicillin G. Cannot get through the outer membrane of the cell wall. And if it can't get through the outer membrane, it can't go in and damage the peptidoglycan that's being formed in the cell wall. And so penicillin G will not work on a gram-negative cell. The gram-negative outer membrane can contain an O polysaccharide, which can help prevent phagocytosis by phagocytes. Phagocytes are white blood cells that come up to a bacteria and then eat them. And the O polysaccharide is actually the antigen in E. coli O157, in this case H7. That's another antigen, but this is an O polysaccharide that gives this cell or this strain of E. coli properties. And O157 and H7 are antigens, but they're also toxic antigens. And this strain of E. coli is a human pathogen. If you were to eat this E. coli and get enough of them in you, you would have gastroenteritis. And I already mentioned that lipid A, a portion of the lipid polysaccharide, is an endotoxin. And I've already mentioned that the porin proteins form channels through this outer membrane that allow molecules to enter the outer membrane if they need to enter the cell. And they also allow molecules to leave the outer membrane. Any question about any of that? All right, comparing the gram-positive cell wall to the gram-negative cell wall. The gram-positive cell wall has a thick peptidoglycan layer, so it's more rigid in its structure. The gram-negative cell wall has a thinner peptidoglycan layer, so it's not as rigid. The gram-positive cell wall has teichoic acid in it. You'll note there is no teichoic acid in the gram-negative cell wall. And then the gram-positive cell wall has no outer membrane of the cell wall. And the gram-negative cell wall has an outer membrane with lipid A, which is a endotoxin. Any question about any of that? All right, let's talk a little bit about the Gram staining mechanism. Crystal violet iodine crystals, the complex, forms in a bacteria cell. In a gram-positive cell, these crystal violet iodine crystals do not leave the thick peptidoglycan layer, so they are retained by the cell as long as you don't over decolorize. And then the cell will stain purple. The gram-negative cell wall has a thinner peptidoglycan layer, and the crystal violet iodine complex wash out of this gram-negative cell wall. The alcohol will then dissolve the outer membrane of the cell wall because the outer membrane is mainly lipid, and it'll dissolve in alcohol. Safranin will then come into the cell and stain the cell pink, giving you the differential color between a gram-negative and a gram-positive cell. This table is showing you the different summary between a gram-positive cell and a gram-negative cell. You can ignore all the lower portions of this table. Gram reaction, the gram-positive cell retains the crystal violet iodine complex. It's washed out of the gram-negative cells. Thick peptidoglycan layer, thin. Teichoic acid present, absent in the gram-negative cell. The periplasmic space is absent in the gram-positive, present in the gram-negative. The outer membrane is absent in the gram-positive, present in the gram-negative. The lipopolysaccharide, including lipid A, there's none in a gram-positive cell. There's high proportions of it in the gram-negative cell. All right, any question about any of that? Now that is the typical cell wall of a bacteria. And there are two types. One, a thick cell wall that we call gram-positive, and then another thinner cell wall that we call gram-negative. And all bacteria essentially follow that differential, meaning they fall into one of those two groups, gram-positive or gram-negative. But there are some prokaryotic cells which are atypical. The first one I'm going to talk about, I should have it first, is the archaea. They differ from the bacterium, so it's not surprising they have different cell walls. They do not have peptidoglycan in their cell wall. Instead, they have pseudomurine if they have a cell wall, and some of the archaea lack a cell wall. Only a few, but there are some archaea that lack a cell wall. And don't ask me what they are because I don't know on the archaea, but I do know that there are some bacteria which lack a cell wall. And these are the bacteria species belonging to the family of Mycoplasma. The Mycoplasmas lack a cell wall, and the Mycoplasma are bacteria. These cells tend to be pleomorphic in their shape because the cell wall is responsible for giving the bacterium its shape, and the mycoplasma lack a cell wall, so they can be any shape that the cell happens to, I don't know, form, be in. The mycoplasmas do have sterols in their plasma membrane. The sterol helps give the plasma membrane strength and support, which is very similar to animal cells, which have cholesterol, which is a sterol, in our plasma membrane, and the cholesterol gives strength and support to our plasma membrane. The mycoplasmas have a sterol, not cholesterol, which does the same thing, and both of them lack a cell wall. The cell membrane is much more fragile than the cell wall, and that's probably why the mycoplasma and the animal cells have the sterols in the cell membrane to give strength and support to the cell membrane. How do you think the mycoplasma stain with the gram stain? Gram negative or gram positive? Well, is the crystal violet iodine complex going to wash out of it? Come on, somebody should guess. I'm just going to move on if nobody guesses. You have a 50-50 chance at the very worst. All right, the third atypical prokaryotic cells with an atypical cell wall is the mycobacterium. The mycobacterium is a bacteria, meaning domain bacteria, but it's a little different. It is a gram-positive cell, but it's a little different because it has acid-fast staining from all of the mycolic acid in the cell wall. Let me see if I have a picture of that. I'm not there. I need to get a picture of that. But I can find one. There we go. Here is the cell wall right here of the mycobacterium. It has a cell membrane shown here, and it does have a layer of peptidoglycan, which is fairly thick, which is shown there. But then it has other molecules in it, the main one being mycolic acid right here. Mycolic acid is a lipid, and all of this mycolic acid in the cell wall prevents water from leaving the cell or water from entering the cell. And that means any molecule that's dissolved in the water will have trouble getting into the cell. And that's why the mycobacterium stain lighter than a normal gram-positive cell. And that's because the gram-positive cell normally allows the crystal violet iodine complex to move right through the cell wall and then stain the cell wall particularly. With all this mycolic acid in the cell wall, the crystal violet iodine has trouble moving through the cell wall. And the mycobacterium, in my experience, always does stain gram-positively, but it stains lighter than another gram-positive cell. And that's because the mycolic acid prevents the crystal violet and the iodine from moving through the cell wall. Any question about any of that? So as you can see, the layer of mycolic acid is actually thicker than the layer of peptidoglycan. And that's why the mycobacterium have an unusual cell wall. They put this mycolic acid in it, which is a very complex structure, and it takes the cell time to make the mycolic acid. And so the mycobacterium tend to grow slower than other bacteria. Oh, I thought I got rid of this one. Good, I haven't logged out totally from here. Let's go back to the lesson. So, three prokaryotic cells, which are atypical. The archaea, it's another domain, so that's not surprising. The mycoplasmas and the mycobacterium. Anyone want to guess on what the mycoplasmas are in the gram stain? Gram negative or gram positive? All right. You can damage the prokaryotic cell wall. I'm not going to go through how you damage the cell wall, but you could digest it with the enzyme lysozyme. You could also damage it with penicillin, like penicillin G. And we're not going to discuss the different names of the cell, like a gram-negative cell with no cell wall is different from a cell wall with a gram-positive cell wall. So you don't really need to know this slide. I'm just telling you that you can damage the prokaryote cell wall. And if you do damage the cell wall, that cell will then be sensitive to osmotic lysis. When penicillin puts a hole in the cell wall, the cytoplasm can protrude out of it. And of course, if you remove the cell wall totally, the cell membrane can expand when water comes in, and eventually the cell will lyse as too much water moves into the cell, breaking the cell membrane. So gram-negative bacteria are not as susceptible to gram-positives to penicillin G because penicillin G cannot get through the outer membrane of the gram-negative cell. But that doesn't mean that gram-negative cells are resistant to treatment. You just need to find an antibiotic that can affect a gram-negative cell. One is streptomycin, and the reason why this can kill a gram-negative cell is streptomycin is a lipid-soluble antibiotic, so it can get right through the outer membrane of the cell wall. But then one of the early members of the penicillin family, I think it was the third member of the family discovered, ampicillin, actually can get through the gram-negative cell wall, and then it can kill both gram-negative and gram-positive cells. So you just need to find an antibiotic that will work on a gram-negative, and there are many of them. Probably about half of the antibiotics work on gram-negatives. The other half work mainly on gram-positives. Any question about any of that? All right, let's talk about the cell membrane, also called the plasma membrane. It is a phospholipid bilayer. So here we see phospholipid. It'll be a bilayer where the fatty acid tails come together in the cell membrane. Let me blow this up. So the bilayer forming here, and there it's shown here, blown up. The phosphate head facing the inside of the cell, which is mostly water, and then the phosphate head facing outside the cell, because outside the cell is mostly water. You can think of that like a blood cell facing mostly water on the outside of the cell. It's this lipid bilayer that actually helps regulate the movement of materials into and out of the cell. If the molecule cannot get through this lipid bilayer, then to either get into the cell or leave the cell, you have to have a transport protein. And there's one transport protein to move each molecule across the cell membrane. So like I said, there are 20 different transport proteins in our cells to move the amino acids, 20 of them, into our cell. Also in the plasma membrane, you can find proteins and carbohydrates. Carbohydrates aren't shown here, but it's something you can find in the cell membrane. There are three types of proteins. There are peripheral proteins, and a peripheral protein is one where the majority of the protein is on one side of the cell membrane or on the other side of the cell membrane. Why it's called the peripheral protein? Because the protein is peripheral to the cell membrane. But there are proteins that span the cell membrane. And these are integral proteins and transmembrane proteins. Here we're looking at an integral protein. Come on. My mouse is dying. There we go. And this one looks like it's forming a channel to allow either molecules to enter the cell or molecules to leave the cell. And there's another integral protein. I'm not sure what that protein is doing because it's not labeled, but it could be a transport protein, meaning a molecule that wants to move into the cell can bind to the protein here, and then the protein will bring it into the cell. Any question about the cell membrane and the different molecules we find in it? If not, let's talk about the membrane and use the fluid mosaic model to describe the cell membrane. The fluid mosaic model states the cell membrane is a fluid that is as liquid as olive oil or as thick as olive oil. And as you know, olive oil is fairly thick. It's much thinner, much thicker than the pouring of like water. You can also distinguish how thick it is when you put it in the refrigerator. Olive oil will become solid, or at least part of the olive oil will become solid in the refrigerator, meaning it'll change from a liquid to a solid. And it's amazing to think about it that my hand is held together by the cell membrane, which is a fluid, a liquid. And that always amazes me. I don't know if it amazes you or not. The cell membrane is a mosaic of molecules, mainly phospholipids and proteins, although there can be carbohydrates in the cell membrane as well. The proteins can move about, or I should say, proteins may move about in the membrane, the cell membrane, to function. Many proteins move about in the cell membrane. Let me blow this up again. The only proteins that do not move about in the cell membrane is a protein either anchored to inside the cell, such as the, what is that called? The cell backbone. Drawing a blank on what that's called. Uh, Intracellular matrix, protein running throughout the cell membrane. Or a protein can be anchored in the cell membrane if the protein is anchored to the extracellular matrix. So think of a bone cell in the bone. The bone is an extracellular matrix and it can anchor a protein in the bone cell. And then that protein is not free to move about in the membrane because it's anchored outside the cell. Any question about any of that? Other proteins are free to move about in the membrane, such as this channel protein. It's not anchored to anything. Come on. So it can move about in the membrane. And those of you who've had AMP, you know that receptor proteins in the cell membrane, they have to be able to move because they typically come together before they're brought into the cell. So if you've had AMP, you should know that proteins may move about in the cell membrane. And it's a liquid, so we'd expect them to move as well. The phospholipids can also move about in the cell membrane. That includes rotating in place, where, come on, mouse, where this phospholipid can actually rotate around right here, but stay in place. But it can also move. Let's say it moves down here, and then probably this nearby phospholipid will move down where that one moved out of. The phospholipids moving about in the cell membrane is actually one of the ways that water can enter a cell. And that is water just waits for the, come on, let's make it a molecule out here where my pointer is. Just waits, let's make it this right here. Just waits for that phospholipid to move, and then the water can come into this gap here. And then it just waits in the cell membrane, this molecule water forming this hole here made by this phospholipid moving, just waits for this phospholipid to move, and then the water can then move across the cell membrane. Now, water can move through the cell membrane, but it doesn't move very fast through the cell membrane. So for roots, for example, in many cells, we have transport protein, which are actually called aquifers, aquaporins, which actually allow water to move through the cell membrane. And you can think of it as a channel protein or a transport protein moving water into the cell. Any question about any of that? All right, so water can move into the cell membrane, but for a root, it doesn't move fast enough into the cell. And so we have channel proteins for the water to move into the cell in a cell in the root of like a green plant. Any question about any of that? All right, the cell membrane shows selective permeability, where the plasma membrane allows some molecules to pass the cell membrane, but it does not allow other molecules to pass. No large molecule can enter a cell without the use of a transport protein. And that's because the large molecule cannot get across the lipid bilayer of the cell membrane. Ions either do not move across the cell membrane, or if they do move, they move very slowly. Lipid-soluble molecules can move right through the cell membrane. And that's because a lipid-soluble molecule can dissolve in the cell membrane and then come into the cell. But if you think about it, like a red blood cell floating around in the blood, most of that blood is water. There's not going to be many lipids in the blood, at least compared to the water. So uh, lipids do move across the cell membrane, but not that often because you don't see lipids coming that often up to a cell membrane and then dissolving across it. But my point is that lipids can move across the cell membrane, which is kind of interesting. So if you were to stick your hand in a bottle of oil, you will eventually start absorbing that oil because the oil will move across the cell membrane. Of course, it may not move through the skin, the dead skin, but it can move through the membrane. Small molecules can move across the cell membrane. The two most important are oxygen and carbon dioxide. Oxygen generally moves into the cell because generally there's higher oxygen outside the cell than inside the cell. And oxygen can diffuse right through the lipid bilayer of the cell membrane. Carbon dioxide is the next small molecule, which is important. Carbon dioxide normally leaves the cell membrane because there's more carbon dioxide in the cell. It generates CO2 than is outside the cell. So small molecules, especially small nonpolar molecules, can move across the cell membrane. Water can move across the cell membrane, but it moves slowly across the cell membrane. And I already discussed that. All right, any question about any of that? Let's talk a little bit about the special cases of the cell membrane. The cell membrane can contain enzymes for various things, such as in a bacteria, in the cell membrane is where ATP production occurs. In a photosynthetic bacteria, this is where the photosynthetic pigments are found in the cell membrane. Now, in the photosynthetic bacterium, let me blow this up again. The cell membrane will invaginate. And in that invagination is where the photosynthetic pigments are found. We call them chromatophores or thylakoids. And here you see the cell membrane. Let me blow this up even further. Get that picture out of the way. The cell membrane doesn't move across like that underneath the cell wall. It actually moves like this. It's a hand, so it's not going to be very precise, but it moves this way and then moves around this fold. And that fold is actually part of the cell membrane. It's a little better with this pointer. So the cell membrane doesn't move like that across the cell wall. It actually moves like this, and then around this fold, then around that fold, and then around this fold. And inside these folds of the cell membrane is where you find the photosynthetic pigments. Oops, wrong one. We'll talk about ATP production later in the term, probably in the next lesson. The plasma membrane can be damaged. And once it gets damaged, either holes will form in it or the plasma membrane will be dissolved, and that will allow leakage of the cytoplasm outside the cell, which will kill the cell. You can damage the plasma membrane with alcohols, quaternary ammonium compounds, and polymyxin B or polymyxin antibiotics. Polymyxin B is one specific polymyxin antibiotic. Polymyxin antibiotics tend to put holes in the cell membrane. Alcohol will totally dissolve the cell membrane. Any question about any of that?
I think you're muted.
How long have I been muted?
Not very long.
Yeah.
Yeah, I don't know how that happened. All right. Please let me know that because I did not see the audio was muted. So if that happens, please let me know. Let's see. And why am I